is Silvia Medrano from the University of Virginia. I'm very excited to be joined today by Dr. Benjamin Humphreys, who is a professor of medicine at Washington University in St. Louis. Dr. Humphreys is an internationally renowned kidney specialist and research scientist. He gave the keynote lecture earlier today, and it was a really very stimulating, very interesting talk. So, Dr. Humphreys, can you tell us a little bit about the topic of your talk?
Yes, and first, Dr. Medrano, let me just say it's a pleasure to be with you here this morning, and thank you for your gracious and kind introduction before my lecture earlier. The topic of my talk was the mechanisms of acute kidney injury to chronic kidney disease, and we've known now for some time that patients that suffer an episode of acute kidney injury are at risk of the future development of chronic kidney disease and even kidney failure. And this is a change from 20 years ago when we thought that if patients recovered, they were fine and just the same as they used to be. And my laboratory has been interested in both the mechanisms of how kidneys successfully repair, but also the mechanisms of acute kidney injury to chronic kidney disease.
Okay, that's very interesting. So, what is that you have mainly learned from these studies, and which questions you haven't answered yet?
Well, thank you. We certainly have lots of questions left to answer, but one of the things that I highlighted in the talk this morning was our use of single cell and multiomic and spatial technologies that allowed us to identify a cell state in proximal tubule cells, an important epithelial cell type in the kidney, that we have called failed repair. And so this is a state that occurs after injury in a minority of cells. Most of them go on to repair successfully, but a small number fail to repair and take on a pro-inflammatory and pro-fibrotic phenotype, which we think may underlie the AKI to CKD transition. And the kinds of questions that we haven't answered yet, but we're interested in answering, include the following. What drives the failed repair state? Is it really responsible for chronic kidney disease? If we were to eliminate failed repair cells, would we eliminate the risk of chronic kidney disease progression? And finally, is there some way that we can turn those failed repair cells back into their healthy counterparts?
Well, that's certainly very, very important. So how can the research that you do can be applied to enhance patient care?
Well, certainly enhancing patient care is the ultimate goal. I'm a physician scientist and I care for patients with acute kidney injury, and so that's one of the reasons why this topic is of great interest to my lab. I think that at this point, what our research has done is identify a cell type that we also see in humans. So we're a mouse lab, but we have replicated our results in humans. And I think that ultimately, if we could develop a therapeutic that would improve this failed repair state, we would hypothesize that that would improve outcomes. It would improve acute outcomes. It would accelerate kidney repair after AKI, and we would hope that it would prevent the risk of chronic kidney disease development. But I have to emphasize that that will all need to be tested in patients, and we're not there yet.
Right. There's a lot of promise, no doubt.
I think so.
Yeah. And where do you see the field of single cell multiomics and spatial transcriptomics moving forward, going ahead?
Well, we're in a sort of golden age of technological developments, as you know, in single cell, spatial, and other kinds of technologies. It's a remarkable time to be a scientist. I think one of the fascinating things is the use of computation and bioinformatics now in science. And the fields are moving so quickly that it's actually very hard to predict where they're going to be in 3 years. But certainly right now, my lab has made a big investment in spatial technologies. And I think the areas where we'll see rapid improvements in the near to medium term future include the resolution of spatial technology. So we already have, in some cases, cellular resolution. I think we will get better and better cellular resolution. And then, of course, I think we'll have that high resolution, but with more and more genes, and ultimately cellular resolution at a genome-wide depth.
Yeah.
And so that's really what's on the horizon.
Yeah. I just would like just to thank you for your time and your wonderful talk.
Well, thank you. I really enjoyed being here at the hypertension meeting, and I thank everyone for inviting me and you for hosting this interview.
Okay, thank you.